Hello students. So we are going to discuss today about Sanger's sequencing method. So the previous method that we have learned, that is Maxam and Gilbert sequencing method, is based on chemical degradation method, wherein by use of chemicals you are basically degrading a specific basis so that you are getting or generating DNA fragments of varying lengths. Okay. Now, here also in Sanger sequencing also, we are going to generate DNA sequences of varying length. But unlike degradation, this is based on synthesis of DNA or polymerization of DNA. Okay. Also, here we can look at the structure of normal deoxynucleotide, this one, which is the building block that is used for replication or for PCR. So basically, the deoxynucleotides have a 5' phosphate group present and a 3' hydroxyl group present. And during extension, a bond formation, one sugar or one nucleotide will be already in place. The extension will take place from 5' to 3' direction. So, to the hydroxyl group of a base that is already in position, the the phosphate group will form a bond, and in this way there is going to be growing nucleotide chain during replication as well as during PCR. Now, instead, if this hydroxyl group is not present, then obviously the extension reaction cannot take place because the phosphodiester bond formation will not take place. So here, what we are doing is we are basically making use of a modified nucleotide, that is dideoxynucleotide instead of deoxynucleotide, which is having a hydrogen instead of hydroxyl group at the 3' end. So as a result of which, if this group gets incorporated during polymerization, then the extension reaction is going to stop, because after this there is no further phosphodiester bond formation is possible. So it will lead to termination of extension, and whenever this dideoxynucleotide molecule gets incorporated, and that will result in formation of varying lengths of DNA or synthesis of varying lengths of DNA. So how do we perform the reactions? So basically, again, we are going to set up 4 sets of reactions. In each tube, you have all the ingredients that are required for a normal PCR. That includes your template, then your polymerase, your building blocks, that is all 4 deoxynucleotides are present. And you use one of the terminating nucleotide, that is dideoxynucleotide, in one reaction. Okay, so in one, if you're using dideoxy ATP, other you're using dideoxy CTP, and so on. So basically, you're going to, depending on the presence of dideoxynucleotide, the reaction is going to get terminated at that particular base when the base gets added. Also, you are using limiting amounts, that is less amount of the dideoxynucleotide. This is because if you're going to use a lot of dideoxynucleotide, then there will be chances that this only will get incorporated all the time. And you will not have varying lengths of fragments that are generated. We want that the dideoxynucleotide gets incorporated randomly at different positions. And only then we are going to generate fragments of varying lengths, as you can see in this example. Okay. Now, next step. So this is just a flow chart for the same thing. So you have your double-stranded DNA. Again, you need to denature it to obtain single-stranded DNA. And then you need to carry out the extension reaction So 4 different extension reactions in 4 different tubes, each containing one of the terminator dideoxynucleotides. So basically, you're going to generate mixture of fragments in each tube that are terminated because of that particular dideoxynucleotide. So at the end, you're going to run your fragments on the gel, and 4 different reactions in 4 different lanes. So obviously all the fragments that are generated here are because of dideoxy adenine. All fragments that are present in this lane, they are because of dideoxy thymine, and so on. So what you basically need to do is, just based on the position of the band, you will know the sequence. You have to just keep reading it. So if I start from the top, it is going to be G, G, A, C, T, G, T, A, G, C, and T. So in this way, I can just, based on the position of the band and in which lane the band is present, I can identify the DNA sequence. Now, this is again another diagram for the same thing. So again, there is a problem with this method is you need to use autoradiography, which is again a lot of radioactivity that needs to be handled. So instead, there is a modification that is done nowadays in which, instead of labeling with radioactivity, you can also label your bases with a fluorescent tag. Okay, so you can have different color fluorescent tags, such that, for example, if it is dideoxy ATP, then it will give a blue fluorescence. So in this way, instead of using radioactivity, I can read the color of the fluorescence, and that way it is a safer alternative. Okay, so nowadays, people make use of this fluorescently labeled dideoxynucleotides for chain termination reaction. And this method is used with the more modern method, which is known as capillary electrophoresis, and that is going to help in automation of the process. Okay, so in capillary electrophoresis, what you have in normal electrophoresis, you're basically running your samples on an agarose gel slab. Okay, while in capillary electrophoresis, you have a tube-like structure basically, and you load your samples at one end of the tube. So the samples will run inside the capillary tube, and let me show you in the next diagram. So here you can see that this is the top of the capillary tube where you will load your samples, and based on their size, they are going to get separated. You have a detection system which consists of a laser beam that is going to make the light incident on your sample, and then based on what base it is, it will give a different type of fluorescence, and that will be read by the detector and will be recorded by the computer system. So instead of like having your reactions in 4 different tubes, you can actually have a reaction in a single tube, where we are going to add limiting amounts of the dideoxynucleotides, all in a single tube can be added, each of them which are labeled with a different dideoxy, a different fluorescent dye. So here, with the help of automation, you can combine the Sanger sequencing method and capillary gel electrophoresis method, so that you will be able to read the sequence automatically with the help of a computer system. So this is basically a chromatogram that is generated in your result or your output file. As you can see, you have these different color peaks that are generated, and they are also of different color. So you can directly read the sequence. Now also it will give you an idea about is the sequencing reaction good. So if you're getting sharp peaks like these, then your sequencing reaction is good. Sometimes what may happen is that in one particular position you will be able to see two different peaks, okay, of different colors. So that means there is some ambiguity in the reading, or there is some error. So you cannot definitely say what base it is. But generally with advances in sequencing methods, these errors can also be rectified. Now coming to the advantages of Sanger sequencing method, you are basically making use of less radioactivity and less toxic chemicals. It is easier to automate by combining it with fluorescently labeled dideoxynucleotides. The cost of the sequencing reaction also is much more affordable. You can have higher accurate reads, which are going to read about as long as 700 base pairs. And commercial kits are also available for Sanger sequencing method, which is quite difficult to generate in case of dideoxy sequencing method. Coming to the disadvantage. So whenever you have your sequencing output, generally the first 15 to 40 bases of the sequence are of poor quality. Poor quality means you're going to have multiple peaks instead of a sharp peak. So you're not very much confident about the sequencing reaction in the initial 15 to 40 bases in your sequencing output file. So this is because of the primer binding and deteriorating quality of the sequencing traces after about 700 to 900 bases. This is time consuming, especially due to requirement of electrophoretic suppression of the fragments. However, with the advancement of capillary electrophoresis, there has been improvement in the time duration that is utilized by the method. Also, you cannot do very long sequences, about 300 to 1000 nucleotide long sequences can be read by making use of Sanger sequencing method, which is better than dideoxy sequencing reads, but still not suitable for whole genome sequencing. So that's it about Sanger sequencing. Thank you.